Hello, everyone, and welcome to today's webinar, 3D Spatial Biology Insights, Simplified with AI. I am Cassie Soltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Microsystems. Leica Microsystems is celebrating its 20th anniversary in super resolution microscopy innovation. To learn more, visit www.leica-microsystems.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Also, if you experience any technical issues, you can utilize the Q&A box and we will assist you directly. I'd like to now welcome our speakers. Yung Choi, Product Owner and Manager, Data and Analysis at Leica Microsystems, and Hoyin, Applications Development Manager, Data and Analysis at Leica Microsystems. Yung Choi, you may now begin your portion of the presentation.
My name is Yung Choi. I'm the Product Owner and Manager for Data and Analysis at Leica Microsystems, responsible for the Aivia product line. It's with great pleasure and excitement that today we're launching Aivia 14 that has a number of discovery tools for 3D spatial biology insights simplified with AI. I'll kick things off with the introduction of the new features of Aivia 14, followed by my colleague Hoyin, who's our Applications Development Manager for Data and Analysis at Leica Microsystems, who'll be demonstrating the complete workflow for 3D multiplex images from segmentation, phenotyping, and data exploration for insight generation. Multiplex spatial biology applications are continuing to grow in popularity, and will continue to see further growth as more and more researchers aim to gain spatial insights for a high number of biomarkers to understand important questions about disease states and therapeutics. In the vast majority of the 20th century, most diagnosis and determination about stages of disease was made by visual inspection by a trained pathologist. With rapid introduction of innovative imaging tools, now scientists can see better than ever with the highest resolution possible. This in return creates a problem for researchers as they can now acquire images that have over a million cells with multiple channels that have essentially millions of data points for which they can no longer rely on simple visual inspection for getting insights. In this example of pancreatic tumor section, we have analyzed a 2D multiplex image with 30 channels and millions of cells with the spatial biology tools available in Aivia. In image A, we have two different types of tumors present in this section. In image B, we used a modified version of CellPost's deep learning algorithm that was optimized for large 2D multiplex images to accurately segment cells. Then in image C, we use an automated clustering method based on the latent algorithm to generate insights, as you see represented here. Here we have two different types of intensity heat maps that are generated based on the types of tumor that we have, and with the rows representing measurements and columns representing clusters. You can see that two types of cancer cells have very different expression of biomarkers. What we are showing you today is a detailed view of the workflow that enables biological researchers to generate deeper insights into their 3D multiplex images powered by AI. More and more studies are showing the benefits of 3D cell modeling even at the tissue preparation or cell preparation stage. As this cancer biology study shows, when you have a monolayer, two-dimensional cell culture versus 3D, the 3D culture shows far better physiological similarity to actual tumor cells in terms of increased cell-to-cell interaction, different exposure to drug molecules, and oxygen-nutrient gradients as you go towards the hypoxic core of the tumor. In addition, when you have cells with highly complex morphological characteristics, as you see in these neurons, the two-dimensional profile, as you see here on the left, does not begin to tell us the complexity of the cells. In addition, when you have a 2D section versus 3D reconstruction, as you see here on the right-hand side, you can get far better and more accurate morphological and spatial insights, as you see in this image that shows a heat map of the 3D distance of mitochondria to the nuclei in sky blue, with the warmer colors showing closer distance. 3D information can help to generate deeper insights in your data, as you see in this last image that was generated using Aivia. Powered by artificial intelligence, researchers can now go beyond appreciating these beautiful images of multiplexed cells here that are acquired by the Leica cell dive imaging system to insight generation without the need to code or train your own models. 6 months ago, with Aivia 13, we introduced a number of spatial biology tools for large two-dimensional multiplex images, starting with visualization of over 100 channels for large 2D images that are multiplexed, to a faster, deep learning-based modified cell post generalist algorithm for large 2D image segmentation. We introduced additionally a number of phenotyping tools that are both supervised, such that users can train the software, and automated, such that users do not need to train. In addition, we introduced a number of interpretation tools, such as this marker cluster dendrogram for spatial insights, Pearson correlation heat map for millions of objects, bin scatter plot for visualization of data distribution for 2 different measurements, and dimensionality reduction for fast exploration of high-dimensional data. So what does a complete workflow for 3D multiplex image analysis look like? An intuitive workflow would include accurate multiplex cell segmentation, phenotyping, and interpretation of data on a single platform with interactive visualization of data so that users can explore different cellular phenotypes in their own spatial context with different cell types or structures of interest. Lastly, users would want to export results to whatever platform they would have in a CSV file format. Here are additional details on the types of insight generation tools that can be utilized in this type of workflow, which we'll explore today in depth. In the solution that we have available at Aivia, users can first define a region of interest and visualize 15 plus channels simultaneously in 3D. There's an option to group biologically relevant channels, which will then become increasingly more relevant as the number of biomarkers for multiplex imaging continue to increase. Then, via a number of different implementation of modified cell pose, we provide robust multiplex cell segmentation powered by artificial intelligence that's deep learning based to handle cells of various morphologies. In addition to our cell pose method, we have additional segmentation methods including automatic neuron tracing, built-in pixel classification, EM segmentation, and cell tracking without the need to code or train your own model. We also enabled all our supervised and unsupervised phenotyping tools to be completely 3D compatible. Once a user has phenotyped and has identified a cluster of interest, additional rounds of phenotyping can be performed using any of our existing phenotyping methods, using any of our available measurements, be that intensity or morphological measurements for further classification. With this secondary and tertiary levels of classification, users can further generate deeper insights into their data. And this can be especially interesting when you can visualize cells that do not belong to any classes that you have predefined. And you can explore them further to better understand what makes these cells different from the training set or user-defined parameters for automatic clustering. In addition, we have a number of interactive data and exploration tools via charting, including this marker cluster dendrogram, a violin plot, scatter plot, histogram, and dimensionality reduction plots. In addition, we have summary stats that are easy to read and interpret for counts and a percentage distribution. Lastly, users can be empowered to make informed decisions about phenotype cells by utilizing confidence mapping. When Aivia does phenotyping, every mesh object or outline object is given a confidence value. That's essentially to sum up how confident Aivia is in that cell's membership into that particular class. There's a preset threshold we have for objects to be classified into a particular class. Users can leverage this confidence value for additional analysis, for example, for quality control for low confidence objects to be removed or retrained, or for high confidence objects to be chosen for further analysis. We have many additional tools that can help you to go from images to insight faster, which we'll be showcasing in a demonstration of the complete workflow. Delving into our 3D multiplex cell detection method, we have modified CellPose, which is a popular generalist deep learning-based cell segmentation method that was published in 2020 Nature Methods paper. There are a few reasons why CellPose is as popular and powerful as it is, and one of the key reasons is because of the diverse training set that the authors use in this paper. In this training set, you can see cells that are round, as you see here, somewhat more elongated, perhaps plant cells, and more bacterial cell shapes, and some more protruding, as you see up here. But they even went as far as to include seashells, garlic bulbs, even road signs to make this training as diverse as possible. As such, the results in CellPose's ability to handle more diverse morphological characteristics of different cell types. In this particular example, I'm showing a result of the multiplex cell detection recipe on 6 membrane markers without nuclear markers. In our discussion with the key opinion leaders in 3D spatial biology, we have seen images very similar to this, where the membrane stains do not overlap, and often there's not any nuclear marker, or if there is one, it may not be universal for all the cell types. As a result, we have built our recipe for applications such as this, where you have no nuclear markers that compete with the rest of your fluorophores and membrane markers that do not overlap. And what you have here is a pretty accurate detection in 2D, then in 3D, and this 3D multiplexed detection recipe based on CellPose can handle images with varying degrees of intensity, shape, and size. This is our fastest deep learning-based 3D segmentation algorithm. We built this tool so that it's compatible not only with multiplex cells, but also with your everyday confocal wide field images as well. So when we test our biomarkers, we can see it's actually faster by 78% compared to our previous method of cell detection. And in addition, we have observed improved segmentation and partitioning in our data, as you see here in these examples. We're very excited about the test results we have seen, so please share with us how you feel about the output of the 3D multiplex cell detection algorithm that we've implemented in Aivia 14 if you'd like to try a trial version of our software. Next, we provide a method to organize biologically relevant channels. So when we look at a panel of antibodies like this, which is provided by Cell Signaling Technology, who's our partner for Leica CellDive, you see a number of markers that are present for different types of leukocytes. In this example, they can be divided into lymphoid, for example, and T cells and B cells. The essence of this feature is that users can now organize these numerous markers under different headers, as you see here, such as T cells and lymphoid cells and B cells, and then visualize them simultaneously and import them into a phenotyping workflow. On the right-hand side, here's an example of a single-plex staining method versus one type of multiplexing method. Markers are being increasingly used together to detect multiple targets simultaneously on a single sample. And the reason why multiplexing is growing in popularity is that when you have a precious sample, be that a clinical sample from a cancer patient or a transgenic animal, you can use that single section to apply multiple biomarkers so that you could see the interaction of those biomarkers in the same cells in the same section. This is a different way than what was done previously in the past, when the scientists are working with a few fluorophores and chromosomes into a single section and trying to decipher the relationship between different sections with different biomarkers and extrapolating their conclusion across different sections. So the way we've implemented this in Aivia is when you have a channel listing, first of all, you notice this grouping on top that's new for us. Users can biologically essentially group any channels and give it a name, in this case, immune cells. Essentially, the way it works is you can turn on all the channels that belong to the biologically relevant channel group in one go for easier visual inspection, and we enable users to import them all into the phenotyper for classification. So the way it works is when you have a phenotyper interface, you can have import group functionality so that in one go, you can automatically create all the phenotype groups with all the channels pre-selected so that the user can start going with training a few examples right away. Next, we enable all our phenotyping methods to be fully 3D compatible. We have 4 types of object classifier options as you see in this interface. For the classic object classifier, users have to select just a few examples, say around 6 for most images, for you to train the classifier, and via machine learning, specifically with a random forest algorithm, all the cells are classified into different phenotypes. We have additional types of classifiers, the phenotyper that was new as of Aivia 13, and 2 additional types of automatic classifiers that I'll go into further detail in the next slides. There are, in addition, a few other options for creating phenotypes using gating tools within the spreadsheet and charting based on measurements and gate by rules as well. We also include a Help Me Choose button to show users which phenotyping option they should use. To sum up, to compare the difference between these 4 types of methods we have, if a user wants to use their expert knowledge about the tissue specimen or cell type they have, by training the software, they should go with the first 2 options, object classifier and the phenotyper. If they want to discover new findings without training examples, for example, in a very highly complex multiplex setup, they can use our automatic classifier, such as the phenograph laden method that we have here. These classifiers have different usage cases. For example, the object classifier ensures every cell belongs to a class, whereas a phenotyper does not, allowing for cells to be unclassified. Also, the phenotyper does not require users to have the exact same measurements for all the classes, meaning a cell can belong to actually more than one class in this setup. For example, a cell can belong to a cancer cell group versus a type of cancer, for example, if there's a specific marker for a specific tumor. On the next couple of slides, I'll go a bit more into details about the automatic methods that I just mentioned that are available within Aivia. The first is a k-means clustering method, which is a well-established, decades-old method that is fairly robust. The only challenge here would be that the user has to know what the right cluster number should be for them to get meaningful data. The way it works is when you have data points as you see here, user has to define the number of target clusters, in this case 4. The next step is random points are chosen as the centroids as you see here. Then all the points that are closest to each centroid are assigned to them to be the member of that class. And the next step is when the centroids are then moved to the average of all the points in that cluster. When that movement of that centroid happens, some of the points may now be closer to another centroid. For example, this orange object here may now be closer to the yellow centroid. In that case, the steps are repeated from step 3 such that the assignment's made to the closest centroid. Then this cluster centroid is moved to the average of the point in the cluster yet again. The steps are repeated from steps 3 and 4 in this iterative fashion until there is no more movement possible. It's a fairly robust algorithm, and the results are usually easy to interpret as you see here. An example with fruits and vegetables in different colors. Let's say a user wants to classify these fruits and vegetables based on the color dimension, so single dimension into 4 classes. You get something very close to what you have here in this kind of rainbow shape with red, yellow, green, and purple classes. But if a user had chosen, say, 3 classes, there might be some surprises. For example, the mango you see here that has a little bit of green and a little bit of yellow might end up in a different class than the user was expecting. This is why choosing the right cluster number is key to k-means clustering being useful. Additionally, this method is also quite sensitive to outliers, which is not the case with the next method, which is a phenograph-laden automatic clustering method. The laden clustering method is growing in popularity in the spatial biology community, such that if you go to talks or workshops, you'll hear more and more researchers using this laden clustering method, which is an improved version compared to the Louvain method. The authors in this paper claim that the laden algorithm yields communities that are guaranteed to be connected, which is a pretty bold claim. To summarize the entire paper into 2 diagrams, imagine you have a region with villages or communities that are round in circles here. Let's call this a red region. As is the case in real life, oftentimes a community may form stronger connection to another community, as you see here with this community that formed a stronger connection to this blue region, and therefore is now a member of that community. And when that shift of membership occurred, the local connection is not updated in the methods such as the Louvain method and other types of clustering methods. What the latent method enables users to have is using a number of refinement and aggregation steps you see here. These poorly connected communities will be forcefully broken apart, and anything remaining in the same region or community will be maintaining a local connection. And what that means for you as a user of this algorithm in Aivia is that when you have highly complex data sets, which is the case oftentimes in multiplex images or cancer biology, you can be assured that this type of poorly connected connection do not stay in the same cluster, and therefore your clusters will represent the best representation of well-connected local communities based on the complex data sets you have. So how to choose between the two? Let's say a user has a cancer sample and the user wants to simply detect cells that are positive for tumor markers versus not. The two classes, healthy versus disease cells, is all the user needs, and k-means will be more than sufficient in that case. It's a small number of classes, and it's very easy and quick to apply k-means clustering based on a single marker. On the other hand, phenograph latent can handle high multiplex data, up to 30 measurements or more. So when you have high dimensionality, we recommend phenograph latent. In addition to the types of classifiers for phenotyping, we have different types of measurements users can use for phenotyping. We use intensity-based phenotyping in 3D, as you see here on the left-hand side, where cells are segmented first, here in the two images right here, and phenotyped using intensity measurements. On the right-hand side, we see an image, not a multiplex image, but a traditional confocal image that we created for our poster at Society for Neuroscience last year. This is an example of purely morphology-based phenotyping in 3D. We took neurons in the granular layer, and based on the characteristics for the dendrites, for example, dendritic branching numbers, dendritic length, and tortuosity, we trained the phenotyper for 3 different levels of arborization, which is the extent of the branches of dendrites you see in each neurons to low, medium, and high. In this particular example, we wanted to understand the limit of the phenotyper, so we actually used only 2 neurons per class. And because the number of measurements that we chose were quite so robust for distinguishing these 3 neuronal cell types with just 2 neurons per class, we were able to get very robust classification in this example. Here's a poster we presented that utilized traditional confocal images, but utilizing all the spatial biology insight generation tools we've created to get to an interesting conclusion about the morphological characteristics of these neurons that we have in this image. If you'd like to receive this poster in high resolution, please visit our website and contact us. Next, we have incorporated an interactive dendrogram for data interpretation. A dendrogram is a very commonly used tool in biology in which closer the distance between the branches, for example, these 2 types of fish, the more similar the types of species or animals are to each other. This is the same principle as a family tree, for example. Your siblings who would be on the same branch would be genetically far more similar to you than a distant cousin who may be several branches away. And this is the same for our dendrogram. We have dendrogram trees for both clusters, as you see at the bottom here, and the trees formed here, which shows which clusters are similar, and for measurements, to show which measurements are similar. Here we have a dendrogram generated from a multiplex 2-dimensional image, but it's exactly the same principle for our 3D multiplex images. You can see immune markers, for example, up here, clustering over here that are on a completely separate branch. Then the tumor markers that you see here. Also, within the tumor marker branches, you see separate branches for different types of tumor markers. This is a very useful tool for users to spatially explore where these clusters may lie and understand the expression of the biomarkers in those cells. These branches actually mean something, such that depending on how you set the parameters, especially for the automatic clustering method, you may achieve, for example, 2 clusters. That could be something like healthy versus diseased cell type, and you can achieve 4 or 7 or more clusters based on the parameters that you have. The number of clusters that can be achieved can depend on where you slice through the tree, essentially. We have improved usability significantly in our dendrogram in this latest release, such that when you select a single tile, as you see here, we have enabled now viewing just those channels in the images immediately. In addition, when you select a row, which is a measurement, you can simply highlight the entire row so that you can easily visualize. And you can also sort, as you see here, both for measurements and cluster, as you see. You can reset any time, as you see right here, so when you want to get the original dendrogram trees back. Next up, we have additional tools here. When you have the objects turned on and you click on the measurement, you can actually visualize a channel. And when you select a single tile, you're seeing the cells in that particular cluster and turn on that channel at the same time. In addition, now we enable users to visualize which channels were used for training. This new table that's on the left-hand side that matches in color to the eventual clusters you generate, so that you can actually group them together and see how well your training markers worked for clustering. There are two different ways to normalize a z-score is represented in the dendrogram in a heat map fashion, and the difference is normalization per measurement option on the left-hand side that allows you to explore per measurement which cluster has the highest intensity and visually inspect the distribution of the markers across all the clusters. When you normalize for the entire table, as you see on the right-hand side here, it's useful for comparing absolute measurements, and you can also do a quick quality control here. When you have, say, a measurement that's consistently low across all channels, that may be throwing off your normalization value, so that you can remove that to rebuild another dendrogram. So both views can be useful for different purposes. When you're working with millions of data points, users also want to get to summary stats quickly. So we built a number of places where you could see summary stats. Number one, we now display by default users to see summary stats in all selected measurements immediately after phenotypes are generated. So you can get total, max, min, mean, mode, standard deviation automatically for every measurement. In addition, we have count measurements immediately displayed, as well as percentage distribution after phenotypes or object sets are generated. These can be exported as a CSV file format for all selected phenotypes, and they can be plotted in a summary stats chart. As I mentioned, we have a confidence value output, which enables users to sort the confidence values that Aivia outputs for every phenotype, and users can choose to select the highest confidence items for further analysis. In this example, we're selecting 0.85 as a threshold and creating a new class, selecting only the highest confidence objects, which is done by gating, and do further analysis from there. In the example on the right-hand side, auto-clustering output is visualized as a heat map with warmer colors representing the highest confidence and colder colors representing the lowest confidence based out of phenotypes. Curiously, the high confidence values are observed along the edge of the tissue, and this could be an interesting finding as well. And users can further explore this by selecting the blue cells with the low confidence values to retrain and update the phenotyper, for example, or users can just select the high confidence cells that you see along the edges for further analysis. After segmentation and phenotyping, confidence mapping can enable you to perform quality control. For instance, you can plot confidence values for different clusters using the violin plot to ensure that all your clusters have a good level of high confidence. You can gate for high confidence objects to create a separate subset. You can further cluster a gated cluster that has the highest confidence or lowest confidence as you choose. Also, using a two-sided violin plot, you can plot confidence and intensity or other morphological measurements in one go. Lastly, I will go into dimensionality reduction to describe why it's useful for handling multiple dimensions of data as you have with high multiplex images. This is a very popular data set known as Fashion MNIST. Data has 70,000 data sample of fashion articles with 784 dimensions. When the UMAT dimensionality reduction was applied to this, what you see here is a large cluster that is separated from all the rest, and this cluster has sneakers, sandals, and ankle boot. Therefore, this is a shoe cluster that's separate from the cluster that's bags over here. This is how you understand essentially dimensionality reduction, which is reducing a lot of dimensions into 2 dimensions so that you can visualize what you're looking for in a separate cluster. What you're looking for is not the color separation, which may show you the phenotype or the class, but good decision boundaries that you see as you see in this separate cluster. What you cannot do with UMAP is, it's important for me to mention, proximity does not mean more similar. Also, the size of the cluster doesn't mean anything in UMAP, and that's very important for me to mention. Here's an export of a white paper by our colleague Vasundhara Agarwal at Leica Microsystems using cancer cells and dimensionality reduction that she's done a fantastic job to explore the complex multidimensional data set that she has and their spatial relationships. This will be shortly available on Leica website. Lastly, I'd like to thank everyone who's worked on this version of Aivia. We have a fantastic engineering team spanning 3 different continents working as a team. Very proud to work with them. Such a delight to work with this fantastic team. And we have a number of other colleagues across all of Leica and numerous external key opinion leaders who were beta testers for this release. Thank you very much for your dedication and insight. Now on to Hoyin Lai, our applications development manager, who will be demonstrating the entire workflow for 3D multiplex image analysis.
And hello, everyone. My name is Hoyin, and today I will be giving you a demo of Aivia 14. Now, a quick word about today's demo dataset. This is a 9-plex human tonsil tissue slide that's been labeled with a panel of 8 OPO dyes and DAPI. This is acquired on a Solaris confocal system courtesy of Tatiana Straka here at Leica Microsystems. On the right, you will see the 3D rendering of this dataset. And on the left, you will see a table of the channel colors, the channel names, as well as the biomarkers and the location and function that they represent. So let's go to Aivia. In Aivia 14, we have designed a user interface to orient around 4 key tasks of image analysis. These tasks form the workflow chevrons you see in the top left corner of the Aivia window. There are 4 chevrons in the workflow: visualization, detection, classification, and exploration. And in each tab, different tools and layouts are available to help you focus on the analysis task at hand. You can modify the GUI layout by clicking on the Options icon and selecting the tools from the menu. You can then save your layout for use in the future. Let us go into 3D view. In Aivia 14, we have revamped our 3D rendering engine to support rendering of more than 4 channels simultaneously. In this demo, I have the tonsil dataset with 9 channels rendered all at once, but we can render even more channels. And here, I am panning around the image and zooming in and out to show you how responsive and fluid the motion can be. On the left above the channel display, we have a new section for grouping biologically relevant channels. These are virtual channel groups for grouping related image channels that can be toggled on and off together, like I am showing you here for the CD3 and CD4 channels. These groups provide a shortcut to changing the rendered display without needing to search for the individual channels in the channel list below. To add a new group, click on the plus icon and define a group name. Since these groups are virtual, the data is not duplicated. You can have multiple groups sharing a set of channels, like I have here with CD3, CD4, and CD8 for this new immune cells group that I'm creating. We will come back to the channel grouping feature later, as there are more workflows that can be unlocked with this feature. In the channel display settings, there is a new button that allows you to expand all of the advanced channel LUT settings. This allows you easier access to the LUT and make adjusting the dynamic range much easier. You can click on the same button again to collapse all of the advanced channel LUT settings. Another new feature in Aivia 14 is the camera sync function for 2D and 3D. Let me click on the sync icon here to turn on the sync. And I have the option to show either a clipping plane or an ortho slicer. I can then use the slider to control the plane position in Z. And I can pan around my image in 3D as usual. And when I go into the 2D view, Aivia will sync the 2D view to the current XY position and Z plane specified in the 3D view. I can do the same in 2D. panning to a different location in XY and navigating to a different Z-plane. And when I switch to 3D view, my 3D rendering will sync to my current position in the 2D view. I am going to my object settings to turn on the meshes object set. The camera sync view will also work with object sets, so this gives me a way to examine my objects in my multiplex image in 2D and 3D. Let's go to a new image. This is a crop of the previous image that we are looking at. It has the biologically relevant channel groups defined already, and as you can see, there are several visible membrane biomarkers for analysis. Another updated feature of Aivia is the new side toolbar next to the image. This toolbar can access the available on-screen tools for the image view in Aivia. And when I mouse over annotations, I can then select one of the tools. This will bring me to the annotations tab. I can use the line tool then to manually measure the diameter of my cell. Now, let us go to the detection tab. The detection tab has a new layout that includes a section for object display and a panel with image enhancement, recipe console, and pixel classifier on the right. In the recipe console, we will go ahead and select the 3D multiplex cell detection recipe, which we are introducing in Aivia 14. This is a cell post-based cell segmentation recipe that allows you to use multiple channels as input. Let us click on the input channel dropdown. For today's analysis, I am going to segment all of the cells with a membrane biomarker on the image. I am going to select CD3, CD4, CD8, and pan-CK as my input channels that correspond to the different immune cells and epithelial cells. In the recipe, I can use the text box or the slider to adjust the detection parameters. I can use the red and green arrow button to select a different processing step from my recipe, like adding a smoothing option or switching between two different partitioning options. And finally, you can click on this button to generate a preview of the recipe output on your image. As I have already defined the optimum parameters for this dataset, I am going to click Start to apply the recipe. While this is running, let me go briefly into the recipe. It takes all the input channels and combine them into a single image input to cell pose for object detection and segmentation. Now, you may be wondering why I should use multi-input channels. In a typical dataset, the staining can be quite heterogeneous, and this presents a challenge for the recipe. Here, I have 3 images, CD3, CD4, and combined CD3 and CD4 image. Let's look at the area highlighted with the red arrow. When we look at the segmentation result from using only CD3 or CD4 channels, we can see both are missing a few cells and poorly segments the cell with the red arrow highlighted. With the combined channel, we can get much better definition of the cell and can improve the segmentation outcome. Additionally, using all of the channels is a major time saver, as I don't need to run the detection recipe on each channel. Once I have all the cells segmented, I can then use an object classifier to separate the different cell types. Here are some best practices for picking the right channel inputs. First, the channel should have low background noise and the staining should be consistent. It should allow you to easily make out the shape of the cell without any breaks in between. Second, the channel should have a similar intensity range. A channel that is significantly brighter than the other channels will wipe out the signal of the other channels and decrease segmentation accuracy. Third, as in the case shown in the previous slide, the channels should complement each other so that you can get as accurate of an image of all cells that are on the image. And finally, avoid channels that are noisy or with low SNR or with splotchy staining. On this image, the yellow channel looks very speckled, and it is difficult to discern the shape of the cell, while the magenta channel has a lot of breaks in the signal. Both issues may be addressed by first using the pixel classifier to enhance the image, then using the pixel classifier result as input to the multiplex cell detection recipe. Let's go back to Aivia 14 to look at the segmentation results. I'm going to move up and down the Z-stack to show you the cell cross-sections in 2D. Let's go into 3D view. But first, I am going to change my mesh color to solid and then go into the 3D view. I am going to toggle the mesh display on and off to show you how closely the segmentation resembles the image. I am also going to toggle the channel display a bit to get a clear view of the mesh objects. Next, let's examine the results more closely. I am going to the clipping plane tool and adding an ortho slicer to the image. I am going to add a clipping cap and make it black so that I can view both the mesh and the underlying fluorescence signal. I will now use the slider to move up and down the Z-plane, and as you can see, the mesh object contour matches up nicely with the image channels. The 3D multiplex detection recipe is not limited to multiplex images. Here, I have a 3-color organoid image with a well-defined cell membrane in the green channel. I have already run the analysis on this image, so I am going to turn on the mesh display and go into 3D. Looking at the results, you can see that the recipe will work just as well with multiplex images and standard fluorescent microscopy images. Additionally, this recipe has the benefit of being even faster and more accurate compared to our 3D cell segmentation recipes in previous versions. Let's go back to the multiplexed image. Now that I have the segmentation of all of my cells, let's identify the cell types. Click on the classification chevron to go to the classification tab. The previous tab with the recipe console is now replaced with the object classifier tab. I am going to select my pre-trained phenotyper from the drop-down menu and apply this to the mesh object set. While this is running, a quick review of the phenotyper. It is composed of multiple machine learning-based object classifiers. You can train the classifier by providing examples of each phenotype. The tool then uses random forests to identify the rules that are most likely to separate the different classes, and it iterates for each phenotype. Let us go into 3D again to see the phenotyper results on the 3D objects. I am going to turn off the orthoplane to show you the whole object. One feature from Aivia 14 that has been extended in 14 is the spotlight function. Right-click on the phenotype group and select Spotlight from the context menu. And now my meshes are colored according to their assigned phenotypes. The phenotyper created 4 classes: CD3, CD4, Pan-CK, CD8, and a new class called Unclassified. The unclassified class are objects that cannot be placed into any of the predefined phenotype classes due to low phenotype confidence. You can examine these objects more closely, and this is a great way to do a quick quality check on your classifier. You can examine the unclassified objects closely and either update your training to incorporate these cells or remove them from your analysis. Let's create a new phenotyper. Click on the Create New Classifier button, and in the window, I am going to type in my new phenotyper as the name of the classifier. In the classification type drop-down menu, I have 3 options: object classifier, automatic classifier, and phenotyper. And if I'm unsure about which classifier type to use, I can click on the Help Me Choose button. This will open a table showing the different types of classifier, their functions, and sample use cases for each classifier. Since I know what I would like to do already, I will pick the phenotyper and mesh to apply the phenotyper to my 3D objects. I am now ready to create the phenotyper. I can choose the Create or the Import Channel Group option, and I'm going to choose the Import Channel Group button. This option will pre-populate the phenotyper with phenotypes that correspond to the channel groupings that we have created. And when I open the measurement dropdown, you can see that the channels that made up each group are reflected in the phenotype measurement channels, as in the case for the CD3, CD4 group with both channels selected. For teaching, I am going to turn on the ortho slicer again and turn on channel rendering as well. Let's start by teaching the CD3, CD4 class. I am holding down the Control key and clicking on the cells to select them. And I'm panning around the entire image to select cells that appear to have CD3 and CD4 signals on them. For the phenotyper, you need as few as 2 cells per phenotype for training, but for a robust phenotyper, I would try to select a few more cells with different signal intensity and morphology around the entire image to make sure I don't miss anything. And when I'm done with selecting the cells, I will right-click on one of the cells and select Assign Objects to CD3, CD4 positive phenotype in the context menu. This put all of the cells I've selected into the training data for the CD3, CD4 phenotype. Now, I'm going to do the same for pan-CK. Typically, you will need at least 5 or 10 cells per phenotype to get a good phenotyper. However, you can update teaching with more cells later if the classifier does not work well for you. Let's finish with training the CD8 positive class. You can use the random forest classifier that the phenotyper creates on another data set in the future. Besides right-click, I can also assign the objects by going to the Assign Objects button at the bottom of the object classifier. Let's select the meshes object set to train and apply the classifier. We can now look at the new phenotypes. I am going to turn off the ortho slicer, and I am going to toggle the object display so you can see how the phenotyper output corresponds to the original image. Next to the subsets in the Objects tab, you will see the count of cells for each phenotype and its percentage of the whole object sets. Switching back to my slides. Here is an example of a larger image with the phenotyper applied. On the left is the raw image, and on the right is the phenotyper output, with 3 classes that we've identified, plus unclassified in gray. In Aivia 14, it is now possible to iterate phenotypes and clustering. Starting from segmentation, go through a first round of classification using the phenotyper or automatic clustering to create my first set of phenotypes. Then, I can create a new phenotyper or automatic clustering classifier. Apply that to one of my phenotypes to create a subcluster of cells. Let's take a look at how this works in Aivia. I am going to spotlight the CD3, CD4 positive set. For cancer biology or drug discovery, one of the analyses is the effectiveness of my immune cells when it comes to fighting infected cells. From the cell state biomarkers, I can make inferences about immunoactivity in the tissue microenvironment. On this dataset, I have 3 cell state biomarkers: PD-L1 and FOXP3 for suppressing immune responses, and Ki-67 for cell proliferation. Both PD-L1 and FOXP3 may be expressed on my CD4 positive cells to indicate immune response regulation, potentially a pro-tumor response due to active suppression of CD4 activities. Let's go back to the object classifier. I will now select the CD4 subcluster classifier from the drop-down menu. This is my predefined automatic classifier using the phenograph-laden method. And when I click on the measurement dropdown, I have the option of using standard or mean intensity measurements or custom, which allows you to select intensity or morphology measurements. I have 4 channels selected: PD-L1, CD4, Ki-67, and FOXP3. Next, I am going to select my CD3, CD4 positive phenotype as my object sets to apply this classifier on. Let's give it a few seconds to run. We will go to the Objects tab again, and let's spotlight the CD4 positive subclusters. And as you can see on the image, I now have 4 classes visible in the CD3, CD4 subset. Now that we have classified our cells based on their biomarker expressions, let's move on to data exploration. Click on the exploration chevron. Now the layout of Aivia is updated again. Let's go to the Charts tab and look at some data exploration options. I am going to select Scatterplot from the drop-down menu, and I'm going to turn off all my other object sets. I can click on the axis label to select the measurement I would like to plot. I am going to plot mean intensity of the CD3 channel against the mean intensity of pan-CK channel. You can see two divergent clusters of data points in the scatter plot. I can use rubber band selection to select a group of cells that have high CD3 mean intensity, and use the gate button in the spreadsheet to create a subset manually. I'm going to deselect the objects and spotlight this subset. With this subset, I can perform additional secondary analysis or go back to my classification tab to create more clusters. Using the scatter plot and rubber band selection to manually gate out objects could be quite useful for those of you working on floral cytometry applications. Let's go back to my previous subclusters. Now, you may be wondering what these clusters mean. For us to understand these clusters, we will need a different tool to examine them. We will go to Charts again and select Dendrogram. In the Dendrogram, I can select the CD4 subclusters as my object container, and then selecting PDL1, Ki67, and FOXP3 to generate a new dendrogram. Here, you see a grid of colorful boxes. Each box is color-coded with the z-score, with red being the highest and blue being the lowest. In practical terms, the z-score is like looking at which clusters have the highest or lowest normalized value for a given measurement. And looking at this, we see 4 clusters emerging from this dataset. The first one is high in FOXP3 and low in everything else. Another that is high in PDL1, one that is low in everything, keeping in mind that the clustering also includes CD4 intensity that is not plotted here, which could mean that this is a conventional helper T cell, and one that is high in Ki67. If I click on the column header, you can see the column on the dendrogram gets highlighted, along with all of the mesh objects on my image. And I can switch between different columns by clicking on the header. I can do the same for rows as well. I'm going to turn off the mesh rendering, so now you can see that as I switch to a different row to highlight, it also changes which channel gets displayed on screen. Finally, you can select one of the tiles, and this will display the channel, and when I turn the object set display back on, the objects associated with that group. I can also sort the dendrogram tiles by right-clicking on the header and selecting ascending or descending to sort the tiles by z-score value. Let's look at my phenotype output for all the cells and select the cell phenotypes as my object container. And I am going to select all the channels except DAPI to generate the new dendrogram. You can see that each cluster also have different intensity levels for the other biomarkers. If I wanted to look at which channel I used for training the phenotyper, I can go to Options and select Show Table. This will show me which measurements are used in teaching each phenotype in the phenotyper. One thing that I didn't show you is that the dendrogram is normalized per measurement. You also have the option to normalize the dendrogram using z-score for the entire table in the Options panel. When you hover over a tile, you will get the z-score value. And look at the tiles, you can potentially infer the interactions between different biomarkers. Take pan-CK, for example. In this cluster, we can see that it is high also in PDL1 and Ki67 mean intensities. In cancer bio applications, we often see pan-CK co-expressed with PDL1 and Ki67. This could be indicative of pro-tumor activities happening in the tissue microenvironment. Let's take our analysis one step further. I am going to spotlight my cell phenotypes group. Say I want to look at the infiltration of immune cells into the epithelium, or in this case, CD3, CD4 positive cells into the pan-CK positive cells. I can go to the spreadsheet and open the Relations tool window. In this window, I am going to create a new relationship called T cells to epithelium. Starting from my CD3, CD4 positive subset, I want to find the nearest object to my pan-CK positive subset. Actually, from the pan-CK positive subset from the cell phenotype set. Then I'm going to click Finish Edit, then Apply Changes. As I am generating the relationship between the CD3, CD4 subset and pan-CK subset, I would like to tell you more about the way the relationship is generated. With Aivia 14, we are measuring the nearest neighbor based on the closest vertex-to-vertex distance between object meshes, as opposed to the centroid of these objects. This gives us a much more accurate representation of the relationship between object sets. Now, what kind of object sets can you create relationships for? Here are a couple of examples. You can go from one object set to another or to itself. You can also go from the second object set back to the first set. One set to a subset, between two different subsets, or one set to a third object set. You can create multiple relationships for a given set, and whatever relations you have specified will also be inherited by all of its subsets. We will now go to summary view in charts to look at the CD4 subcluster's relationship to PAN-CK. I am going to plot the mean nearest object distance, then turn off all the other object sets besides the 4 CD4 subclusters. The error bar is quite large on this image, so it doesn't appear that I have anything statistically significant about the difference in mean distance between the CD4 positive cells and the epithelium. Though it would appear that cluster 4 has an ever so slightly smaller nearest object distance. So with a larger image, this may be an interesting hypothesis to test whether this type of CD4 cells from cluster 4 may be influenced by the proximity to the epithelium, using the measurements that are available in Aivia. Let's look at the intensity distribution of the CD4 channel for these subclusters. In charts, go to violin plot, and I'm going to select mean intensity of CD4 on my axis. The bulge from the violin plot represents the data distribution. And in the center, you have a dot and a bar that represents the mean and standard deviations respectively. When I hover over the plots, you can also see a p-value bar above the chart to indicate the statistical significance between each group. Here is another new feature in Aivia 14. I am going to right-click on the chart and select two-sided violin. This will give me a second violin plot where I can plot, for example, mean CD4 intensity with the nearest object distance. Now I have two distributions, CD4 intensity on the left, object distance on the right. We have other chart options that I will not be going over today. These include dimensionality reduction and Pearson's heat map. With Pearson's heat map, you can look at the Pearson's correlation coefficient between different measurements. And finally, you can use the export function in the spreadsheet or in the charts to export the data to Excel for further analysis. Let's go back to the visualization tab to examine the image more closely. Another new feature of Aivia 14 is color by measurements. I am going to select my meshes object set and at the very bottom, change the color setting to color by measurements. Once again, I will use the nearest object distance and change the color scheme to a different rainbow spectrum. Then spotlighting the PAN-CK positive subset, you can see where the PAN-CK cells are and where the CD3, CD4 cells are, and the relative distance between them, with red being the closest and blue being the furthest away. Another way to use this feature is for quality control. I will now plot the CD4 subcluster confidence. What this shows me is the confidence assigned to each object that is placed into a cluster. I can look more closely at the cells that are red and orange in this view. These are my lowest confidence value cells. And using the tools at my disposal, I can then decide whether these cells are misclassified, in which case I can either update my object classifier or remove them from analysis. This is a great way to verify my analysis. To finish off this video, I am going to zoom out to look at my whole image and show you how to create a video animation for your lab presentation or share with your colleagues. Go to the Video Animator tab. There's a new feature called Create Sample Animation. Click on that, and you can see the animation timeline is now populated with many keyframes. Let's play the sequence. This option lets you set up a video sequence for export quickly without defining your own keyframes. Today, we have covered many of the new features of Aivia 14. We showed you the new workflow-oriented user interface. We talked about cell segmentation using 3D multiplex cell detection recipe. We covered the process of phenotyping and cell classification. And we explored different ways to chart and display our data and perform analysis on an image. And with that, I would like to thank you for your time and move on to the Q&A. Thank you.
All right. Thank you, Won-Young and Hoyin, for your informative presentations. We will now begin the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. Let's go ahead and get started. Our first question says, I am a development biologist working on Drosophila embryo development, and I do not have multiplexed images or work with cancer cells. Can I still use the phenotyping and data interpretation tools you presented?
Thank you, Cassie. Can you hear me okay?
Yes, I can hear you perfectly.
Awesome. It's a fantastic question. In our design philosophy, we wanted to make sure that this tool we were creating for Aivia 14 was applicable for most biological scientists, even those who do not do multiplex imaging. So we currently have a complete workflow that can work for people who are doing imaging with a single channel, typical 4 channels in a comparable image. Even with bright field images, you could phenotype using the same workflow that we have. And we have a number of deep learning models, as well as different segmentation models, such as our neuron tracing model that can handle various types of images. And the best thing we can do is get your data and see whether we can do it for you. And then you can decide whether it's a good workflow. So please contact us and we can show you what it looks like, what the solution will look like.
Wonderful, thank you. All right, our next question that we have here says, I work with collaborators in other institutes. Is it possible for me to share my parameters, and what options are available for me to just share the results with them?
Fantastic question also. So I'll take this one away as well. So we have a free Aivia community that can be distributed with any of your colleagues. It can be installed on any PC workstation and anywhere in the world. And when you finish analyzing your data, your collaborators can open your analyzed images using the community, or Aivia community. So that's one solution we can have. And in terms of sharing parameters, we have different ways of sharing parameter settings at different stages of the workflow, that if your collaborator has a full version of Aivia or a subscription license, then they can actually just apply your parameters to their images and to essentially run the experiment using the same parameter settings. So we can help you with that if you need help, and we can show you what's possible.
Wonderful, thank you so much. And it looks like we have time to squeeze in just one more question. That question says, I only work with histological sections currently and would like to know if you have any solutions for non-fluorescent images.
Yeah, that's a great question. So we do have a number of different tools that are capable for doing H&E staining and other bright field applications. So for example, let's say we have the pixel classifier, which allows you to provide a few examples of data sets or images and cells that you would like us to detect, and the software will automatically determine what is a good cell to identify and segment that as well. And then with the segmentation, you can also run this through all of our phenotyping and automatic classifier to generate the results that you're looking at.
Excellent. Thank you again, Won-Young and Hoyin, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Leica Microsystems, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time. Take care, everyone. Goodbye.
Thank you. Bye-bye.
Thank you, bye.